My name is Michael Maguire. I'm CEO of Avectas, a company which has developed a next generation ex vivo cell engineering platform called Solupoor. So we've heard several presentations and panels today talking about the onward march of cell therapy and the need for cell engineering. In fact, disruptive new methods of cell engineering, which enable all of the new things that therapeutic developers want to do. These include delivery of cargoes, mRNA, protein, DNA, gene editing platforms, and new emerging tools. Interestingly, combinations of all of these as well. These have diverse applications in gene editing, CAR-T or CAR-NK, CAR+, which is a term I can't claim to have coined, but we've certainly heard on the road, and that's combinations of gene editing and CAR therapy, and of course iPS products as well. So sophisticated engineering approaches and sophisticated tools to enable those are needed, and Solupoor is central to those. So as in such a fast-moving space, the future is in some respects here already. So we've seen recent IND approvals for combination products which have a viral transduction step and also have an electroporation or a non-viral step as well. So we see viral delivery has been relevant to CARs, TCRs, etc., and non-viral delivery doing a range of additional functions to the cell in order to condition it for when it returns and does its therapeutic job within the body. Moving even beyond the next generation, we see the very alluring possibility with a lot of the challenges around viral transduction and access to virus manufacturing around virus and so forth, the possibility of an entirely non-viral approach to produce persisting or enduring changes to cells is very exciting and something which potentially Solupoor could enable. So those are some of the things that a technology needs to do. The cells which are produced from that technology need to be fit to go back into the body to do their therapeutic job. And the body can be a hostile place for an engineered cell to go back into. So this could be in autologous therapies. This could be because the patient material in the first place was fragile. It could be in allogeneic setting, it could be healthy donor material. However, when the cells are reintroduced into the body, they're under multiple attack, if you like. And so what's needed is that the cells which are engineered are in the best possible condition going back into the body, and the best possible condition is guaranteed by the gentlest possible technology, which will minimally perturb the cells, but effectively deliver the cargo into the cells and enable them to do their job. So our solution to that conundrum, if you like, or that big wish list of asks is Solupoor. And Solupoor is a combination of a device and a chemistry which come together to engineer cells. It exists in two formats. The first is a development and optimization tool, which we call Flexi, and it operates at a scale of about 10 to the 5 cells. And then that scales to our disposable single-use device called MIDI. And MIDI operates region 10 to the 8 cells with a big brother or big sister, if you like, achieving 10 to the 9 cells or beyond. This development addresses the needs of GMP alignment, flows with the market in terms of disposability, and will enable developers to do their optimization work on one platform and scale almost seamlessly to a scaled platform. And this in-development platform is currently being validated, scaled and differentiated by us. So in terms of a a quick tour of some of the data which we're getting from this platform. First of all, delivering GFP. We're delivering to naive T cells, naive CD3 positive T cells, and also to T cells which have been activated in different ways. We're getting pleasing GFP expression. We've also delivered larger constructs. So for instance here, an editing tool, which is between 4 and 5 times larger than a GFP mRNA, and it has a tag on it as well. We've effected gene editing in human primary T cells across a number of platforms, and there are 6 or 7 gene editing platforms out there. We've effected gene editing on 4 of them with excellent results, which form a beachhead or a baseline upon which we can optimize and move forward. The type of targets that we're addressing in the gene editing are the type of targets involved in immuno-oncology, which you will be familiar with from the literature. We've also synthesized our own mRNA CAR cells. And in addition to that, and most recently, in conjunction with a company who are happy to be disclosed, Applied DNA Sciences in New York, we've been delivering linear DNA, and we've been delivering it with very pleasing efficiency. Once again, a beachhead and a point from which it can be optimized. What's striking here in this data is that the performance of the Solupoor on the viability of cells which are delivered is significantly enhanced over electroporation. Speaking further to that enhancement over the other available technologies, proliferation is a great catch-all indicator of functionality. And we can see in this graph here that solupurated cells, which have been expanded over 7 days, are unchanged from unengineered cells, however, are different from electroporated cells. This data holds for fresh material and for freeze-thaw material. In addition to that, we introduced solupurated cells into a xenograph mouse model. And there's 2 interesting things about this data here. First of all, in the untreated group of cells here, we're looking at front and side scatter on the flow cytometer, and we can see the population of cells just after transfection with the for Solupoor and for untreated, solupurated and untreated cells. And we can see that the population granularity is the same, if you like. The nucleoaffected cells, which were done at the same hour, in the same hour looked at, the population has changed there, and that's interesting, as these cells went on to fail to engraft into the mouse in the model that we described. We've done this, generated this data ourselves, but we've seen similar data as well from other groups working in this area. So in terms of the business, Avectas is firstly committed to progressing and deepening our current external feasibility studies. So we're working with a number of therapeutic developers in the United States and research institutes in the United States and in the UK. So managing, deepening, and growing those is something which is close to our hearts. Establishing new collaborations with cell therapy developers is something that we want to do. So as the need for sophisticated cell engineering develops, we want to be there to seed our technology early with institutes developing those next generation of products, or with therapeutic developers working on those next generation of products as well. We want to establish a key partnership to conduct a first-in-human study. We want to do that. We have a very aggressive timeline for doing that during 2020. So In the words, we've a fantastic scientific advisory board, and at one of our inaugural meetings with the advisory board, the chair said, if you can countenance electroporating cells, you can countenance soluporating cells. So I've taken that as something which I think myself and my team would like to bring soluporated cells into a therapeutic program that we can dose a patient with them. We're also interested in looking at other synergistic possibilities in such an exciting and fast-moving space. I'm continually surprised, I suppose I shouldn't be, but I am continually surprised at the level of imagination, sophistication, and just the energy in the field, and we want to continue to be part of that. So finally, in terms of the developmental timeline and the key milestones which we need to hit to stay on track, we have our internal validation of the MIDI unit, which I showed you. In the third quarter, we are transferring that technology into 2 sites, one in Canada, one in North America. In Q3, we will be doing a range of in vitro killing work using soluparated cells and with a partner as well. And in Q4, our single-use device will be available to us for validation. Moving into 2020, our single-use system will be internally validated, and finally we will position ourselves for a first-in-human dosing with a partner. So I'd like to thank you for listening. I'm delighted to have been able to share an overview of Soluporation technology and Avectas with you, and good luck with the rest of the conference. Thank you.